Next speaker is David McCormick, Chair and Director of the Institute of Neuroscience at the University of Oregon. We'll speak on the brain is a precise functional connectivity machine. Need some help?
Great. Good afternoon. Happy to be here. When I met Nikos the first time, it was like the first time I went on the internet. There's so much information all at once. We have a saying in the U.S., it's like trying to drink water from a fire hose. He's just obviously a brilliant man, lots and lots of ideas. I know he's going to be missed here, but I look forward to interacting with him in the future and exchanging these interesting ideas. So today I wanted to talk about one of those interesting ideas because we were asked to talk about a big topic. And I thought a big topic would be, what is the brain? And my little description here is it's a precise functional connectivity machine. Now, I moved to Oregon 2 years ago from Yale. And after moving to Oregon, I became actually kind of obsessed with climate change. And I've been talking to the leadership at SFN. Your flight to SFN next month, if you take economy class, is going to generate about 0.4 tons of CO2. That's calculated from Stuttgart to Chicago. And you can compensate that if you want by planting about 60 trees for a year. If you want to do one tree for 60 years, that's okay. Or you can go on this German website and you can donate to their cause and they'll help offset your carbon load. I plan on doing the same or not flying at all. So, yeah, single neuronal responses are highly variable. They're noisy. This is a mainstay of neuroscience. This is the topic I'm going to talk about today. And this was a big topic a decade ago. It's a little less so now, but I wanted to resurrect it a little bit. And so the noise of variability is a measure of, and you don't have to vote for this, but just think in your head, is this a true measure of the variability of signaling in the brain? Or is it really a measure of our ignorance of how the brain works? Right? And you can know from my title what I think about that. So the brain undergoes large, medium, and small scale variations over time scales, ranging from hours to milliseconds. And I used to study sleep and waking. This is my grandson when he was born a couple years ago. Sleep and waking, and now I'm actually more interested in rapid changes that occur during waking state. I call this seminar behavior. You know, you might be very attentive and very on at one moment, but at the next moment you're off in space and you're not paying any attention, and all of a sudden you're back again. All right? These are the kind of rapid changes that I'm very interested in understanding what the physiological mechanisms are. B.U. He and I are writing a review right now for Annual Review of Neuroscience on this topic, and I wanted to mention it real quick because Nikos brought it up. What is a state? So this is how we imagine state. Behavioral state, you know, in the behavior is some kind of large measure, but also nested, so you could be sleep or you could be awake, and during awake you could be aroused. If you're aroused, you could be still or you could be moving. And if you're moving, you could be walking, well, not probably trotting and galloping, but certainly a horse or a dog can do this, you know. And these are all kind of nested states. And then if you measure behavior, you're measuring different things like locomotion, pupil diameter, your facial movements, and so on. And they vary or co-vary over time. Maybe you're a neuroscientist, so you measure brain activity, you read or the EEG, FFT, or multi-unit activity. And then you plot all these things on multidimensional space. And what we imagine is that your brain wanders around in this multidimensional space that has as many dimensions, as many things as you'd like to measure. Some of them are relevant, some may not be relevant. But you have clusters of places that you revisit. It's not random, and it's not uniform, and there's some disallowed places, and there's some allowed places. And your brain wanders around, and your behavior wanders around inside that space so that you go through it in a kind of a somewhat non-random, kind of orderly manner. And that's what we mean by state, is those revisited places in that multidimensional space. How many states are there? Well, I was reading my grandson, I showed a children's book, and they had emotions for every letter of the alphabet. Here's some emotions for the letter A, and these are all distinct states. You can be acceptance, adoration, affection, agitation, alarm, alertness, alienation, and so on. And you go to letter B and C. So there's a multitude of emotional states as well as other kind of states. As a neuroscientist, we're very, very poor at talking about the neural mechanisms of these states, which are obviously very important for an average person. So we have to look at kind of very general features, and I'm going to talk about a couple of those general features here today. All right? So Yang Dan already presented this, but when you're deep asleep, you have these slow waves in your cortical activity, up and down states, and you might have spindle waves and so on. And then when you come to waking, all that kind of tends to go away. You still have some of it, and you're more in an activated state. Right? Those are the big state changes for sleep and waking. But even during waking, there are these rapid changes in state. I mentioned seminar behavior, but that have an effect on how you respond. And this curve was proposed by Yerkes and Dotson in 1908, so over 110 years ago. And they proposed that your ability to do something has a sweet spot, a Goldilocks zone, in which you have your state just in the right position. So you may not be too aroused or too stressed. You may not be too unaroused or drowsy. You're in this correct spot. So, and you may be wandering around on this inverted U curve on a regular basis. So how do we study that? We study that in a mouse. We have head-fixed mice, and we head-fix them so that we can do all the cool things you can do, intracellular recordings, imaging, and so on. And we ask the mouse to do a task. In this task, the mouse is asked to report whether there's a pure tone in the sound or not, and the pure tone is embedded in noise. So it's a bit of a difficult task, and we vary the level of the tone so that the animal has to pay attention and be engaged to do the task. All right? And this was work done by Matt McGinley here. All right, I'm going to show you a video of a mouse doing the task, and I want you to watch the mouse. And there's lots of things to watch, actually. You can watch the pupil of the mouse. You can watch the eye position. You can watch the whiskers. You can watch the nose. Mice can move their nose in very complicated ways, or the licking of the mouse, and so on. So he's asked to lick if he hears the tone. And now the mouse is sitting here. He's kind of drowsy. He's waking up. You see the pupil this is in real time, by the way. And the pupil got bigger, and now it's getting bigger. All right? It's medium-sized now, but it's getting smaller all of a sudden. All right? Over about a second or two. Now it's getting bigger. And the animal's licking, it's getting bigger. And he's getting actually a little over-aroused now, maybe from all the rewards he's gotten or from running. His legs are probably running. We videotape that now, too. And his pupil is quite big, but now it's got smaller again. Okay, those changes on that time scale are very relevant to behavior and brain state. Changes on a time scale, I would say, is on the order of fractions of a second are relevant to understanding what's going on in the brain. All right? Now, when we saw this a few years ago and noticed that the pupil diameter was very important, we did what all good neuroscientists we do. We go on YouTube and search pupil diameter and got this video here. Do I have sound on this? Because I'm going to play this and see. Yeah, okay. This is a cat watching a horror movie. You can watch their pupils of this cat. It's getting quite concerned now. Something's coming in.
Mrs. Bates.
Okay, good. So when we were doing this, we measured the hippocampal activity because hippocampal activity is a good indicator of state. We also videotaped the mouse and looked at pupil diameter. These are sharp waves in the hippocampus. And then we plotted the rate of sharp waves in the hippocampus with pupil diameter. And you can see that the sharp wave rate here in black and pupil diameter follow each other quite remarkably. All right, and this is 20 seconds. And here's the animals walking, and you can see the pupils dilate and the ripple rate decreases. And then even if the animal is just sitting there, he's not walking, the pupil diameter and sharp wave rate are going in lockstep together. So the state of the whole animal, hippocampus, cortex, pupil, is changing on a second-to-second basis. And the same thing is happening in you and I, and that's why I call it seminar behavior, all right? It gets even weirder. We did whole cell recordings in cortical neurons in the auditory cortex, and in some neurons, not every neuron, but you would see things like this, which are still really remarkable correlations. You would see that every time the pupil dilated, we call these microarousals, the membrane potential of the cortical cell would depolarize. In between those depolarizations may be slow oscillatory activity, but then these microarousals, which were not associated with walking, were associated with these depolarizations of the cortex. And when the animal decided to go for a walk, there was a predictable sequence of events in which the membrane potential of the cortex would depolarize and the pupil would dilate, and then the animal would walk. And then when the animal stopped walking, the opposite would happen. The membrane potential would come down, the pupil would come down, and the animal had stopped walking already. All right? And then you would go back to having these microarousals, which are on a time scale of a couple of seconds each. And these have very big effects on the cortical activity and the animal's ability to do the task. So here you see the microarousals here, and here on the depolarizations in the cortical network that are occurring with that. So we looked at the behavior of the mouse, and it seems very, very noisy, right? Here's the animal doing the task. Green is he got it right. Red is he licked when he shouldn't have. And blue is he didn't lick when he should have. But then if you look at the pupil diameter over the 2 or 3 hours of the mouse doing this task, you see that it's varying all over the place. And the black dashed line actually is the optimal state for the animal to do the task. So if you sort the data by pupil diameter, what you see is exactly the Yerkes-Dodson curve. If the pupil is very small, the animal does poorly on the performance. This is hit rate. And if the pupil gets bigger, the animal is getting a little more aroused. The hit rate goes way up, and then it goes back down again as arousal gets more high. This is a very high level of arousal here. So this is the Yerkes-Dodson curve. And remember, if I go back to what's happening in the animal, these variations, which are occurring on a second-to-second basis, are having a big effect on whether they can animal can do the task or not. It actually has a big effect on what's going on in the brain. That's, well, I'm sorry. This is, I was surprised to find out my data is extra big, extra significant here, you know, on the news the other day. It's all drawn by a Sharpie. This is a full data set. Just trust me, I'm going to skip past this. It's true for all the animals. I got a dog at the same time as this. I was wondering, well, why does the animal have to be in a very particular state to do a detection task? I took the dog to the veterinarian, and whenever the vet would walk by, she would freeze, hold her ears, because they have these mobile pinna at the bottom of the floor. And yeah, she would freeze and just stop breathing. So then I looked in the animal literature, and it's actually a common thing for detection, auditory detection. Animals tend to do freezing behavior, and they stop breathing, and they try to stop generating any semblance of self-motion. All right? So we had trained the mice to do an auditory detection task, and we found that they did it best if they were sitting perfectly still in a semi-aroused state. If they were running, or if they were very aroused or drowsy, they did it poorly. So what's the neurophysiological basis of that? So here's a whole cell recording in the auditory cortex of a mouse. And the pupil is kind of intermediate diameter here, and we're presenting complex sounds to the mouse over and over again randomly. And these are the synaptic response in this auditory cortical cell. These are synaptic responses. And here is a particular sound when the pupil is kind of intermediate. This would be the best state for doing the task. And here's a robust response. But then when the animal was walking and the pupil got quite dilated, we gave the same sound here, number 2, and you can see the response is quite different. If I overlay them, the big difference between the two responses is that there was a lot of spontaneous activity even without us presenting a sound. It could be that the mouse made a little sound itself, the claws on the wheel or something like that. The wheel itself doesn't make any sound that we know of. Or it could be just some movements of the ears or self-generated sound that was causing this, but it, I think, degraded the signal-to-noise ratio of the animal to be in this highly aroused state because of the spontaneous activity that was going on in the mouse. All right? So here's the point of the precise connectivity. Does this spontaneous activity, what is its use? You know, when the animal changes state, when he moves, or we've heard talks about this, you know, about corollary discharge and so on. I believe that this changing the state of the mouse, when what they're attentive to and what they're doing, is changing the gain of the neurons by depolarizing them and changing the input-output curve of the neurons to connect and disconnect different groups of cells so that you can make information flow the way you want it to. All right, that's the big picture theory. So we looked at the gain of the neurons. We looked at their input-output curve with whole cell recordings or with multi-unit recordings, and we found exactly that's the case. The gain of the neurons, be the firing rate or evoked PSPs or the reliability of the cells, was an inverted U of pupil diameter. So the animal was optimizing the gain of their cells, of the cells, in the auditory cortex by getting in the right state, by being still and attentive and aroused, and not too aroused. All right? We measured lots of different things in the brain and in the behavior. Here's the behavior in red, and the brain activities in green and blue and black, and we found that they all were optimal around the same zone of arousal. All right? So it's kind of this, and the reason that behavior in the brain seemed so variable was because the animal was varying in his state on a rapid basis every second to every second. Right? So I usually get the question, why not just get in that optimal state and stay there? Why not just be in that perfect zone and stay right in that perfect zone? All right? So I searched YouTube for the answer again. I got this video. These are two gazelles that are fighting. All right? They're totally focused. These guys are just watching. All right? And this is the bad consequences of being totally focused. These guys say, okay, I'm getting out of here. Because they're not noticing anything, right? Okay, well, you know what's going to happen now. That's the end of that. So, well, you don't want to be in that zone. So I searched YouTube again. What do you want to be in the zone? And I came up with this video, Nico the Rabbit. But I think we should call it Nikos the Rabbit in honor of our guest here. All right, Nikos the Rabbit. Nikos is smart. Yeah, he's not going to be totally in the zone. He knows what to do. He knows how to deal with danger. Okay, Nikos, run. Run, Nikos. There comes the eagle. Go, Nikos. Oh, yes. All right. Yeah, good. We're going to see that one more time in slow motion here. This is great. He's coming. Where is it? Here you go. Nice. Nikos escaped. I'm not going to say who the eagle was. Okay, so, all right, so why is the brain doing this? Well, we can ask the physiological mechanisms. Pupil diameter is a cue. What controls pupil diameter? Well, you know about the parasympathetic and sympathetic pathways. And this is a kind of a fictitious diagram that's in all the textbooks, but it's proposed that the locus coeruleus somehow controls this pathway and controls pupil diameter. All right? So we looked at cholinergic fibers and neuroadrenergic fibers in the brain of mice, and we looked at how their activity, this is with two-photon microscopy with Jacob Reimer and Andreas Tolles, kind of a collaboration with Nikos secondhand, I guess, because Andreas was in your lab, right? With pupil diameter. And what you see is that pupil diameter actually tells you a lot not only about the state of the mouse, but the cholinergic and neuroadrenergic fibers. So here's a raw trace of a cholinergic fiber in orange and the pupil diameter in black and walking in green. And you can see that when the animal got a little bit of a pupil dilation with a little bit of movement, the cholinergic fiber discharged, all right? It's a very good correlation there. And this is true for neuroadrenergic fibers too. So the pupil, and these are the microarousals. These only last a couple of seconds. And every one of these little dilations of the pupil were associated with a burst of activity in the neuroadrenergic fiber here. And if you do a kind of average pupil dilation constriction cycle of these microdilations, these 2 or 3 second little microarousals, you could see that the cholinergic fibers discharge. In anticipation of that, the pupil is a smooth muscle. It's delayed by about half to 3/4 of a second. And the neuroadrenergic fibers also fire in anticipation of that. And if you look at the coherence between pupil and neuroadrenergic fibers or cholinergic fibers, you see very high values. At 0.3 hertz, for example, the neuroadrenergic fibers are of about coherence of 0.6 to 0.7 between pupil diameter and neuroadrenergic, any random neuroadrenergic fiber on average. So that means the neuroadrenergic fibers and the cholinergic fibers should also be highly correlated with each other in the mouse brain. So Lindsay Collins in my lab generated this whole mouse brain window so that we can image fibers over the entire mouse brain in awake state. This is called a crystal skull. It's bent like the skull and replace the skull with glass. We put them into a mesoscope, and then we can image axons here, here, over the mouse brain with the two-photon microscope. It has a very large objective. And you can record these axons, and this is 20 times sped up, but you'll see them blinking, okay, as the animal behaves. And if you look at this activity, so here's an example of 3 neuroadrenergic axons in the animal's walking, pupil diameter, and whisker movements here. Turns out the face is really, really important to understand what the mouse is doing. You see that the 3 axons that we are plotting here seem to be highly correlated over this long period of time. And we zoom in on a little bit of it, and here's whisker movements. This is 2-second time base, and you see all 3 neuroadrenergic fibers, even though they're about 1 millimeter apart, are highly correlated or doing something together with movements of the animal's face. 15 minutes left. This is correlations between neuroadrenergic fibers with each other and cholinergic fibers with each other with distance across the brain 4 millimeters is about half the distance from the back of the brain to the front of the brain in a mouse. The mouse brain is about 9 millimeters in front to back, not counting the olfactory bulb. And you see that the correlation is quite high. This is 11,000 data points, so it's a lot of data, and it's about 0.5, and it's about 0.7 for cholinergic fibers, and it does fall off a little bit with distance, but not a lot. Now, I don't know whether how much of this is the same axon innervating different parts of the cortex. We're trying to work on that now. This can't all be the same axon. We didn't image one axon. It's many different mice, for example. All right? Well, if you look at human, when we first saw this, I wrote right away to Marcus Rakel and others. I said, you got to look at this in human. Maybe this explains, signal in the human. And in fact, Yellen and others published this paper in which they looked at, let me go back. They looked at the default mode network and in other networks, and they found that some networks were highly correlated with pupil diameter. And here's an example of activity in the default mode network in red, and here's activity, there's the pupil size, all right? And this is in occipital networks, and this is pupil size, all right? So something's going on that's kind of universal. So we wanted to do our own fMRI study, and this is coming out this month in cerebral cortex. But we didn't do fMRI, we did a little bit better. We did GCamp6 wide field imaging. So David Salkoff, a grad student, trained mice to look for a flash, a 50 millisecond flash, and to report that flash with a lick, and he degraded the intensity of the flash to a point where the animal was making mistakes. He was getting hit rate of about 65%, so that we could get misses also. All right, I'm going to show you a video here. There's a lot going on. This is in real time, but I want to show you that we have two videos, one of the animal's face and one of the animal's brain from top. This is occipital cortex, this is motor cortex, this is somatosensory cortex, retrosplenial cortex. All right, so the animal was licking there, got a reward. This is the running speed encoded here. All right. So let's see. Distractor, that was a tone that was presented. The animal's whisking. Okay, he got a sight, and he licked and got a reward. All right, it's hard to keep track of all this at once. You have to kind of sit and watch it for hours. All right. But you see the brain activity, there's a lot going on, right? How much of that brain activity can we explain? It's not all just pupil diameter. It doesn't all go up and down. In fact, pupil diameter only explains about 10% of what's going on here. All right. Okay, so let's look at the brain maps and see if we can figure out what's going on. All right. So this is just average activity delta F over F for one mouse. This is for many mice. This is the flash-evoked visual cortical response. This is somatosensory body area. This is motor cortex. This is generated by the movements of the mouse and the responding. And this is especially generated by licking up here. And we know all that from the data I'll show you. And this is the probability of activation during a trial of the brain from top. We don't see the sides of the brain. This is just the dorsal surface. This is the time to activate. So early on is visual cortex, and then a little bit later you get motor and somatosensory areas activated in preparation and during the movements of the animal. And this is the grand average of 10 animals in that data set, all right? So secondary motor cortex is important in performance of the tasks. So here's a HIT trial, and you see this nice spot of data in the motor cortex, and in the MISS trial, the visual cortex is lit up, but the animal didn't respond, and so the motor and sensory areas don't really activate as you would perfectly expected. And this is true in all the mice here, all right? The choice probability, if you look at pixel by pixel, how much does that pixel's activity predict the choice of the animal? The choice of the animal to lick or not lick. And you end up seeing there was a nice spot here in the area that Carol Svoboda studies a lot called ALM. It's in secondary motor cortex. If you stimulate this part of motor cortex, it makes the animal lick, even in naive mice. So this has something to do with performing licks, all right? And so this predicts early on whether the animal is going to lick. And I'm not saying it's where the decision is made, but it's kind of where the motor plan is or the motor movement is generated for the response, all right? And here's an example of that. So this is that spot, and this is the activity when the animal made a hit. And here they the mouse licked spontaneously, and you see that motor cortical area is activated. All right, and this is a correlation between that motor cortical area and lick rate, and you can see a nice correlation there. So this part of the cortex seems to have something to do with generating licking, whether it's spontaneous or learned responses. And so we put mucimol into that part of the cortex to see if it was critical for the animal to respond. And mucimol activates GABA A receptors and blocks activity. And sure enough, the hit rate went down, the false alarm rate went up, so the D prime of the animal went down significantly with mucimol. All right, so that the discriminability went down. All right, so now this is the interesting data. All right. So then we gave the data to a computer scientist and said, can you predict, her name's Erin McCarthy, can you predict the activity in the brain by the behavior of the mouse? And this was inspired by work done by Matteo Carandini and Ann Churchland. And so we gave the computer scientist a video of the mouse, and then the data about the trial, like what kind of trial was it? Was it an auditory trial? Was it a light trial where they're supposed to lick? Was it a catch trial? Was the light intense or dim? And so on. And then we gave data about what the choice of the animal made. Did the animal hit? Did it false alarm? Did it miss? And at what time did that? And then the person knew the time, and then they measured things about the video. Very simple things. The first simple thing, and the most important one, was just what's the motion energy, what's the change from frame to frame that occurred in the gray level in the movie, pixel by pixel, and added up to one number. And then what's the change that occurred in the whisker pad area? What was the eye position? What was the pupil diameter? All right? What was the running speed? And then took these 10 different parameters and ran it through a ridge regression linear model system in the computer, and then gave it new data and asked the computer to predict the new data. So this is the prediction by the computer. The black is the actual data, and the gray is the prediction by the computer of activity in secondary motor cortex, activity in somatosensory cortex, activity in visual cortex. And here's some of the actual behavior, okay? And you could predict a lot of activity in all these different brain areas just by knowing these factors. None of these are brain-derived. They're all behavior-derived from the video, right? And now we show, this is the student that did this work, we show the total variance of the dorsal cortex of the mouse that we can explain by those 10 variables. And we can explain about 60% at the peak of the brain activity as viewed from a wide field. It's not single cell, it's just kind of a gamush of many different things. About 60% just by looking at those factors. What are the most important factors? Well, video motion energy in the face was the most important factor. That's shown in red. It could explain by itself about 35% of the variance of the brain activity. Whisker motion energy was next. Walking or not, choice, eye movements, and so on. So you see movement is the biggest factors. Face movements, whisker movements, walking, choice, licking or not, eye movements were the biggest factors in determining what was going on in this dorsal cortical view. Right? Then you could plot each factor. You could plot the beta weights on space of each factor. So whisker movement told you a lot about barrel cortex, somatosensory cortex, motor cortex. Eye movements told you a lot about occipital cortex. Choice, whether the animal licked or not or missed, so on, told you a lot about secondary motor cortex. All right. So Carandini's lab published a paper in Science just recently that recorded 10,000 neurons in the mouse while the mouse was spontaneously behaving. And then they sorted the neurons by likeness to each other. And here's running of the mouse, and this is pupil diameter of the mouse, and this is whisking of the mouse. And then this is the 10,000 neurons. And you can see a certain fraction of neurons, maybe a third or so, are highly correlated with these movement-related events. This is in visual cortex. So about a third or so of the neurons were highly positively correlated. A bunch of neurons were negatively correlated, and some neurons seemed to not really care that movements of the mouse, and especially the whisker movements or the running of the mouse, was encoded in these visual cortical neurons. And I think that's where we're seeing also in our wide field is that movement turns out to be a really important signal in mice. All right? So Dennis Nestvogel, I have just a couple minutes left, did whole cell recordings. He's a German. Hopefully he'll be coming back to Germany to do this kind of thing. Did whole cell recordings in visual cortical neurons. And he tracked whether the animal was whisking or not, green, and whether the animal was walking or not, purple, and then the pupil diameter in red. I'm just going to zip ahead to this. This is whisker movement. This is membrane potential. Look at that. You know, it's crazy, right? Green is the whisker movement, and black is membrane potential of a pyramidal cell and visual cortex, all right? And this is walking. And so you can even make a computer model and have the computer model predict the membrane potential of a pyramidal cell and visual cortex based on the video of the face of the animal and whether the animal is running or not. And the computer model did a pretty good explanation of the basic membrane potential of the visual cortical neuron. All right. My last slide. So, what does this mean? It means that state is king. All right? State is king. It explains at least half of the activity in even the visual cortex. So if you're not measuring state of your mouse, or maybe even of your person, you're not measuring half the activity. If I wrote to a grant and said, I want to study visual cortex, but I don't know what visual stimuli I'm giving, they would think I'm crazy. I'd say the same thing. If you're studying visual cortex and you don't know the state of your animal, you're doing exactly the same thing. You don't have half the data, all right? There's something driving these systems, the cortical systems, the modulatory systems, the thalamic input. What that something is, I don't know. Maybe it's mesocephalic neurons, giganocellular neurons that are arousal-based. They're driving everything in unison to change the state of the mouse together, and it's causing a very big but decodable, decipherable signal in the brain. And the brain, therefore, is not as mysterious as we thought. It's more precise. You just have to look deeper and find out where that signal is coming from. Thank you.
Okay, thank you. Time for some questions.
Thank you, David, for brilliant
I
talk.
don't think the microphone's on.
It's not on? It's on?
Oh, okay.
I just said thank you for a brilliant talk. That was important to say in the microphone. But this issue was already alluded to yesterday, I think.
Yeah.
All of these experiments that study perception or any other function in different sensory cortices or motor function always use reward and punishment to train the animals. So this is with rarely any control for the impact of the reward or punishment on the behavioral state. So how can we get around that if you just want to study pure perception, sensory processes, for example?
Well, a lot of our mice are also not untrained, although we habituate them. I think a bigger factor for mouse research, maybe other animal research, is the head fixation and the time of day, the state of the mouse, because mice, we reverse cycle them, so they're during their active period, but still their propensity to sleep varies over that period, right? And so we put them in a very unusual situation. They can't take naps. and they're in there for a couple of hours. So they're head fixed. They can't curl up in a ball, take a little nap, and then run around and do what they would do. So they're kind of stuck in this zone, like sleep driving. You got to keep the car on the road, but you want to take a nap, And so I feel like sometimes they're caught between these two worlds, and that's perhaps why we're seeing these dramatic changes. Now, that being said, I do think that all people studying mice should be carefully looking at their state, and like you said, the reward associations, and also just their sleep-wake state. Yeah.
Oksana?
Thank you. It was a true pleasure to listen. If I may say canonical in this audience. So canonically, we think that state regulation is bottom-up, but
Right.
how do you think, how strong is top-down control of the state, especially in the context of cognitive tasks?
Well, we know that top-down is very, very important. You can change your pupil diameter even by seeing a picture or seeing someone and you're poof, So your state is also very top-down, and it'd be very interesting to examine those pathways, and we hope to do that. In the mice, I'm not 100% sure how top-down it is. It'd be interesting to try and to work on that. I think we're seeing a very much bottom-up mechanism going on, but I'm sure there's some top-down.
Last
Tony?
question,
Yeah.
Tony?
Yeah, David, I'm interested broadly in the question of it's one you've just really been talking about, which is how general conclusions from the mouse are, especially for primates, because people like Josh Gold and others have documented many of these same changes on perhaps a smaller scale. And I'm wondering if you just had to guess and spitball, what do you think the best way to index this stuff is in primates, and how important are some of these whole brain-like signals going to turn out to be?
Yeah, I worry a lot about working on mice. I've worked on different species. I've never worked on primates, but mice are very. They're kind of ADHD, right? They're very reactive, and they don't. They're whisk. What's going on? Like this all the time. Maybe as humans we learn to suppress that and be more just focused. But so I worry a lot that what we're seeing is exaggerated in mice, but the fMRI data suggests that it's there to some degree in humans. So I agree with you that in monkeys and humans, it may not be half the activity, but maybe 10% or something. But it could still be driving a lot of variability. That's a good point. Mice may be very attuned to movement, right? But it'd be a good discussion point, yeah.
Okay, we have a coffee break. Thank you very much.